So yeah, so I manage the day-to-day operation in the Genomic Sequencing Center that you have seen this morning. I have another hat, which is a cancer center. I don't know the name, it's a long title, but basically my job is to advise and help everyone in the cancer center here at Cold Spring Harbor to use those technology because we typically have two types of users. We have very big lab that do thousands of samples a year, and obviously I don't have much to teach them, they know how to do it. And we have labs that do a few or maybe 10 or 20 samples a year. So my job is to help them design experiments, work with them, see what can be done, cannot be done, what are the best choices, and make sure that their samples go through the system in a timely manner. I also actually do some research or manage some research projects for the DEED Microbe lab, mostly in plant genomics, de novo assembly of plant genomes, as well as in colon cancer. We have a collaboration with Stony Brook. I was at the Jetson lab before, where I got about the same kind of job, where I actually created their sequencing facility. And before that, I was a faculty member at the University of Missouri in Columbia. Go Mizzou. If you follow college football a little bit, and basketball. So anyway, my email is here, and those guys, Mona has my email as well. So feel free to send me any question following the course. I'm more than happy to answer them, okay? So, piece of introduction first. One of the biggest issues with RNA-seq is that there are no canned way to do it, okay? There are many different choices that you can make. All of them will impact the data and the analysis downstream, okay? And the choices you are going to have to make are mostly dependent on four things. First of all, the organism you are working with. How big is the genome? How many genes are expressed? What's the fold change of expression in that particular organism? The other one is obviously how much money you have. It does matter, and how much time you have, okay? Also, if what type of viability, can you get the sample easily or not, right? Sometimes you are limited by the type of sample you have, sometimes you are not. But bottom line, and most important of all, the biological question you're asking. For example, I'm going to compare a normal versus a tumor cell. I can guarantee you there are lots of genes that are differentially expressed, and a lot of genes are widely differentially expressed between the two cells, okay? So I don't have to go very deep to find differences. In my previous life at the University of Missouri, I was actually, part of my research was looking at heat stress, chronic, long-term, mild heat stress on mammals, on mice and on cattle. Not the type of heat stress you read about in the books, that's basically a sublethal type of heat stress, Basically, you do that for more than a few hours and, the yeast or whatever else die. Talking about, 15, 20, 30 days exposure. So it's kind of a mild stress, but it has a lot of application in the real world. And obviously, the differences in expression are not very big, right? You're not killing the organism, but you are putting it in a different type of environment. That was a lot more challenging, okay? Because we had to dig a lot deeper to find small differences. So those kind of things are going to direct, and nobody can make those choices but you, because you're the one who knows what you want to do, right, at the end. So it's up to you, but I can hopefully give you some idea of the type of choices you have to make, and give you some ideas of what happens if you do take one or another, okay? So that's really what I want to do. There are no magic recipe in RNA-seq. Okay. So the first thing to do is that, as I like to say very poetically, shit in, shit out. Okay? Sorry, but that's true. And that's never as true as in RNA-seq. Okay? The first thing you have to start from is good RNA. It's huge. If you don't start with good RNA, you should not even try to spend your money. Okay? Don't even do. So RNA extraction is the first one. You probably all have all kind of different. I don't have any stock in any company, by the way. Okay, anyway. So you can pick up whatever favorite kit you have. But personally, especially when I was at the Jetson lab, where we had extracted over 40 or 50 different RNA from 50 different tissues from the same mice, we were essentially using the Trizol Plus because I found out that it was really a very versatile type of kit that really worked every single time, okay? So I loved it. But again, you can choose any kit that work. I'm just giving you some recommendation. It does work. It does not work, however, for cell lines or for very small things like a mouse adrenal gland. Yeah, we did that too. That was fun. But if you ever try to dissect an adrenal gland from a mice. But for those type of things, we went to the RNA-easy, RNA plus kit, okay? She's much better for cell lines, again, or for those very, very small tissue that you can't really homogenize very well and see very well, okay? So that's my two recommendation. But again, as long as you have something that works, it's good. The biggest thing is this. So you can just run your RNA on an agar-house gel to look at it, but let's face it, it doesn't really help you very much to see the quality of it most of the time. The best thing to do is to run it on an Agilent bioanalyzer. Now, I'm fully aware that some RNA, like some of you guys had NSET RNA that really look different on the bioanalyzer than everything I've seen before. That's fine, it happens. But typically on an RNA, on an Agilent bioanalyzer, if you run your RNA and you get those kind of profile here, okay, the size of your RNA and some measure of intensity, it's just an intensity here, okay. It returns to you a number. It's called a RIN number, for RNA integrity number, and you can go to the website. There is a lengthy explanation on how they calculate it. But basically, what the software does, it look at the peak at the 28th peak, at the 18th peak. You look at the ratio of that, which is easy, but you also look at how much DNA is between those two and how much DNA is here, okay? And what you really want to see is something too nice peak and very little between those two and here, okay? Because the 18S and the 20S in the eukaryotic genome are obviously 90+% of your RNA, right? This is actually, by the way, those are the small ribosomal RNA. So it gives you a number from 1 to 10. Anything between 8 and 10, by experience, is usually good for RNA-seq, okay? This is a profile of a 9.1, this is an 8.1, and you can see you're starting to have more peaks here and here, which is an indication that some of your RNA start to be degraded, but not too much, okay? I've been as low as 7 on some tissue, the pancreatic tissue, for example, which is really hard to extract RNA from, okay? And you can see that at 7.4, you're starting to see a much jagged lines with a lot more degradation, and those ratios are changing as well. I will never go under 7, to be honest. I won't even, because what happens is that your RNA get degraded, okay? But degradation doesn't happen randomly, okay? Some RNA degrade faster than others. Some 5' RNA, some 5' are degraded faster than others. And so now if you look at differential expression, what you are going to do is you are going to do, you don't know if what you see is your biological, biology, or if it is just consequences of the differential degradation between samples, right? So basically you draw conclusion not knowing if it has any meaning. So I would definitely try to get it above 8 and definitely never go under 7, okay? You can see I get to 6, you basically almost have lost almost your 28S completely, a little bit of 18S. For some reason, actually, it's weird, but the 28S seems to degrade faster than the 18. Again, I was talking about bias. That's one of those. I don't know why. So if you don't have one, send it to a place that has one, okay? Because you're going to spend a lot of money, you better make sure that your RNA is worth it. If you don't have a lot of RNA, so you can make library now easily from down to 100 nanogram of total RNA. I'm talking eukaryotic type total RNA. Can probably, easily go down to maybe even 50 with the same kit. When it gets under that, it gets more difficult. Okay? And it gets to the picogram range, then you need to basically be able to amplify your RNA. And that's just an example of, for that company, it's a small company in California, has a lot of kits, but they have really made their name on RNA and DNA amplification. Okay? They use what they call it's not their SPIA system here. To basically work, by the way, they have a special primer, which is half DNA and half RNA, basically. And what they do is that they do, if you go here, their hybridizers are primer to the cDNA, start a synthesis, and then bring up an RNase H, which basically degrade the primer, go away, and then you can bring another primer which start to displace the double-stranded DNA and start again. Okay? Work really well, by the way. It works really well. There are other systems out there, rolling circle and stuff like that. Personally, that's the one I will definitely recommend, the NuGen product, if you have to amplify RNA. Okay? By the way, if you don't understand my accent, you'll tell me. I'll continue in French. I know that one of you can understand. So, to be or not to be? That's the first question you have to ask yourself. What do you want to look at the end, in the analysis? There is really two types of differential expression analysis you can do. One is to take all the sequences that you get back, map them against a gene model. Okay, so your gene model tells you from base A to base B, this is where my gene is, everything that mapped there is part of that gene. And you just take that number as a measure of expression. That's a gene level expression. You don't try to differentiate between the different isoforms. You don't care. Or you can do a transcript or so-called isoform differential expression. So now you want to know if your condition A versus condition B, isoform A is up or down, and isoform B is up or down, and so on. Okay? The biggest complication here is that obviously, since your potential target, your potential number of sequences you want to measure expression from now is, it's much bigger than just a number of genes, then you have to get a lot more sequence. That's where the cost issue comes from. Okay? Because you are counting something. Okay? So counting things that are expressed very high level is no problem. Everybody can count globin or whatever, or actin. Okay? But if you are trying to count something that is expressed at mid-level or low level, then you need a large number of count, right? Because you can't compare condition 1 and condition 2 if you have 2 counts in condition 1 and 3 counts in condition 2, You need numbers. So you need to sequence deeper. Costs more money. Now, there is other thing that came here too. First of all, sequence length. So you can sequence a variety of different lengths, right, in those platforms, from typically 36 to 100 on a high-seq, you can go up to 300 bases on a MiSeq. On an edge-seq, you can go up to 150 bases. Okay. Anything above 75 is fine for an RNA-seq. You don't have to go very high. Okay. The question is that 75 in an eukaryotic genome is basically the limit where you can pretty much map most of what you are going to map anyway. Most of what is unique in a genome will map with a 75 base pair. Okay. You can do 200, you can maybe a couple of percentage more that will map and so on, but really 75 or 100 is the limit. You're not going to get a lot more mapping if you have 150 bases. Again, I'm talking human genome at that point. Okay. The other thing is pan versus single RAND, and that goes with those two, with this one. If you want to do gene level expression, single RAND is fine. Single RAND read, which means you get one read per molecule of cDNA, per molecule of original RNA, right? Pan is you get 2 reads. You get a read from one side of your cDNA molecule, and you get another read from the other side of the cDNA molecule, right? Now, careful, a lot of people made that distinction, but forget to make that distinction. But when you do pan read, you are still counting the same number of cDNA molecule. You get 2 reads per cDNA molecule instead of 1, but you still counted 1 cDNA molecule. And therefore, you're still counting the same number of RNA molecules you started from. Okay? To remember that. Okay. So why per hand? Because if you want to do transcript differential expression, you need per hand, because the software that are going to take those reads and try to reconstruct the different isoforms that are expressed from your gene, they need that per hand information. They are really tuned to use this. Okay? They really do not perform well. They don't perform very well to start from, but they really don't perform very well at all if you have single read. Okay? So it's up to you. From the beginning, you have to ask yourself, do I'm only interested on the gene level or the transcript level? If I'm interested in the transcript level, you have to do a per hand. Okay? And at least a per hand 75. Per hand 100, whatever. Which brings me to the other thing, the software that does this. So, you know, what happened is that you get 100 bases read, right? You get 2 reads or 1 read from the same molecule, it doesn't matter. You feed that into a software, like, you know, the Tuxedo suite of software or others out there. And you know, software spit out, oh yeah, I found that is, you know, 3 isoform for that gene. I found 2 isoforms. But he's trying to reconstruct things how? By looking at those reads and trying to find exon-exon boundary, right? In 100 bases, okay? Typically on 100 bases, you might find 1 exon boundary. You very rarely find 2, right? So it's a lot of computation behind the screen to try to take all those 100 bases read that come from your gene that may have 3, 4, or 5 isoforms and try to piece them together. By the way, the isoforms are not expressed at the same level now. So you don't have the same number of reads that cross the same exon boundary depending on the expression of the isoform. So it's really complicated, and those software try to do the best they can. But I think that the consensus now, at least from people working at ENCODE, you guys know what the ENCODE project is? Okay. Is that the software that do that don't really work very well at all. Okay. At least seems to be the consensus that 2 years ago where people were saying oh, it's okay, and now it's not really okay. Okay. I mean, we do miss a lot of things. So there are a lot of false negative and there are a lot of false positive. And it's kind of natural, you know, out of 100 base pair and 1 exon boundary, you are going to have problems reconstructing your whole construct, your whole transcript. There are limitations there. That's where the longer read can help, by the way. Okay? Because now you can hopefully cross maybe 2 exon boundaries. So, in other words, the consensus, at least on the people who do that for a living, seems to be shifting away from those transcript analysis because essentially it's really difficult to do the analysis. That doesn't mean that what you get is bad. You can get very useful information from those. But you have to be really aware that you have a lot of false negative and a lot of false positive. So you are going to have to do a lot of validation in the back end to ensure that what you see is true. And you have to know that you are going to miss a lot of stuff. But even if you find something, if you can validate it, that's good, right? So it's up to you, really. You have to make those choices early on, okay? Another choice you have to make is the type of RNA-seq library preparation you want to do. Now, that's essentially stranded RNA-seq versus normal, okay? So what happened is, you know, you went through that with micro polyase selection, you fragment, you random prime, do a first cDNA selection, a cellular cDNA selection. You ATAL, you put your adapter at the end. This is the regular stuff, okay? So you do a double-stranded DNA like you will do from any kid that does a double-stranded DNA out there, okay? The stranded RNA-seq, what they do is that when you do the second strand of cDNA, you actually use DUTP and not DTTP, okay? And then, depending on the kit, you either use a glycotransferase, an enzyme, to shoo away, just before the PCR, to shoo away the second strand of the cDNA. Or in the case of Illumina, they use a polymerase that basically is not going to use that DUTP as a it's going to stop at the DUTP when it tries to actually copy that strand, okay? So either way, either you shoo it up or you don't amplify it. But what you end up at the end after your amplification is only the first strand of DNA, right? Which is a mirror image of your RNA molecule. It's directly, so now you're only reading the strand, all the sequence you're going to get are only going to be from the strand that the gene is on, not from the complementary strand, okay? So you're only getting sequence from one strand of the DNA now. Okay, the strand that contains the gene. Why is that important? Because one of the things that the Genome Project and the ENCODE project has shown is that there are quite a bit of genes, at least in human and higher mammals, that have overlapping fragments. There are microsatellites everywhere, long RNA that are transcribed, okay? So it's not because you have a gene on one strand of the DNA that you don't have meaningful transcription on the other side, on the other strand. Now, if you do a regular RNA-seq, you get sequences from both of those strands. You don't know from which one it comes. You don't know if it comes from your long RNA or from your gene, right? You have no way to know. But if you do a stranded, you know. you will know from which strand it comes from. So you know how many reads come from the strand that contain a gene, and how many reads come from the long RNA, which is transcribed in the other side. Okay? So now you can have a much more powerful analysis. And that's typically an example of, I think it was Drosophila, different embryo larva stages of Drosophila. And this is a regular, this is a gene model, by the way, okay? It's the gene model. And, you got different time series experiment, and, you have different expression, and they saw that, that thing coming up, and they say, oh, annotation, apparently it's a new exon, right? Annotation doesn't show an exon here, but it's a very clear expression here that, appeared 1-2 day larva and so on, and continues later. It's a new exon for that gene. Well, when they did a stranded RNA, what they found is this. It's actually expressed from the other strand. It's not a new exon from that gene. It's something else. Okay? And it's probably meaningful because it's actually, highly regulated. It only comes at a specific developmental stage. Okay? So most of the suppliers now have a stranded RNA-seq kit. They are a little bit more expensive, typically 30%, 25% more. Buy one. Okay? It doesn't make sense in my mind to do a regular RNA-seq anymore. Okay? They're a lot more difficult. It's the same type of product. It's just use DUTP instead of TTP. Okay? So yeah, buy stranded. Okay? Okay. Polyacylation versus ribosomal RNA depletion. So what we did for your sample and what Michael did is use the stranded mRNA prep kit from Illumina. Okay? mRNA means it's actually doing a polyacylation. So you take your total RNA, okay, and you actually pull out all the RNA that have a poly A by using a poly T probe. Okay? By priming with a poly T, when you do your cDNA. Okay, that's fine. It's classic, there's nothing difficult. Everybody has done that. You can also do something else, which is to actually deplete it in ribosomal now. Instead of selecting only for the poly A, you deplete, you remove the ribosomal RNA, and you take everything else. So those technologies are called ribosomal RNA depletion. Illumina has some kit like that. They're called, the things they call them total RNA, whatever. Every company has a different name, okay? But basically what happened is that they use a different trick, again, but they remove the ribosomal RNA from your sample. What is left is everything else. Everything that has a poly A tail and everything that does not have a poly A tail. Okay.
Just briefly, what's the trick?
So there are different tricks. Some of them will actually do a cDNA synthesis with non so random primers. That is, primers that are specifically designed not to bind to a ribosomal RNA. Okay, that's usually the most kits are working that way. Okay, and obviously now you have different kits. So you have kits for mouse, human, and rat, and you have kits for plant because ribosomal RNA have different sequences, right? You got some kits for bacterial RNA, gram-plus, gram-positive, gram-negative. Okay. It doesn't work, obviously, 100%, but it's enough to remove most of the ribosomal RNA from it. Okay.
Could you use magnetic capture with ribosomal RNA specific?
Yeah, it's complicated. Most kits went away from it. It's another way. Early on, they were trying to do that, yes, but it ended up being a complicated system. Basically, it's just more easy to put a bunch of primer into the solution and do a cDNA synthesis. When you do ribosomal RNA versus poly A, you catch more stuff. When you remove the ribosomal, whatever is left behind could have a poly A tail. It could be a long RNA that doesn't have a poly A tail, right? So now the potential population of RNA that you can sequence is actually bigger if you do ribosomal depletion instead of a poly A selection. Okay. So if you are interested in long RNA, for example, that's definitely the way to go. You're going to lose most of them if you do a poly A. Okay. Also, if your RNA is degraded and you can't get anything else, biopsy, for example. Hey, you got a biopsy, you have a biopsy. You don't get another biopsy. That's what you have, right? So, and it's degraded. Well, poly A is going to be a problem because it's going to prime from a free prime end and then stop once, where the RNA is broken. since the ribosomal RNA usually do a not-so-random priming across your RNA length now, you'll be able to create cDNA from everything, even if it is broken down into 2 or 3 basic pieces. So you'll be able to catch more of the 5' end, okay? Versus a regular one. Here we see the poly A, where you can see start dipping pretty much, when you get to the last 20, 30% 5' end, okay? So you get a better coverage. Well, that's a pro. The cons, actually I should have put that here. It's seriously more expensive. The kit from Illumina is twice more expensive if you want to do, okay? So we are talking $3,000 versus $6,000 to do 48 samples. It's not negligible, okay? So again, go back to how do you decide what you want to do? Well, cost is obviously a big one, right? And again, if you're only interested in the protein coding regions, you don't really need to do ribosomal RNA, okay? You can do a poly A, it works great, okay? So again, you go back to what do you want to study? What is your experimental design? What are you interested in, okay? Nobody can make the choice but you. Okay, number of reads, that's one I get all the time. Number of reads. Short question, I don't really know. Okay, but there are some guidelines out there. Okay. So there is a nice review this year published in February by Slim and al. In Nature Review. It's not only on RNA-seq, it's on ChIP-seq, it's on exome sequencing, I mean the whole seq biosphere, if I may say. But it's a good review, so I definitely point you to that one. Okay. In terms of RNA-seq, they actually cite that work from the ENCODE consortium. They actually took a cell line, so now it's human cell line, and they sequenced up to 214 million. So they really went very deep. I mean, nobody sequenced 200 million but the ENCODE consortium. And they then sample down, right? So if I take, 9 million out of those 214, if I do that 1,000 times, and I look at it and say, okay, if I look at the expression level of my genes, how many of those genes can I detail the expression level within 10% of what I get when I get to 214? Okay, so basically, as I said, 214 is my true level expression, which might be true, but, well, okay. At least that's the best I can do. If I do 9, how much of that do I recover instead of doing 214, right? So, and then they broke it out by how highly expressed is that RNA. So anything above 1,000, that's actin, that's a retrotransposon, that's something that's very highly expressed in a cell, 1,000 copies per cell, sorry. 100 to 2,000, those are usually abundant RNA. 10 to 100 are considered mildly abundant in the mid-range of the expression level. 1 to 10, that gets to be really rare. Okay, rarely expressed RNA, and less than 1, it's even worse, okay? Per cell, 1 RNA per cell, okay? As a sideline, well, I'll talk about that later, but I can't say it now. There is some good data out there that show that at least in humans, the range of expression of RNA is probably 6 order of magnitude or so, okay? To give you an idea of what you have to look at from between the most abundant and the least abundant RNA. Okay, so if you do the 36, you can see that you recover 80% of your RNA measurement are within 10% of the true value, okay? For everything except for the very rare one. If you want to go to the very rare one, you probably have to go to 80 million reads, okay? So it all, again, depends on what you want to study, right? If you want to study something that is expressed on the red curve, you can't go away with 10 million. You want to study something that is on the greens, or if you want to catch most of it because you don't really know what you're looking at, well, you probably have to go to, things like 70 if you can afford it. That's the other thing, if you're going to pay for it, all right? So again, there is no set rules. You all depend on what you want to do. It's up, it's up to you, but it's a guideline at least. I give you some ideas, okay? Right now, we do for the, my advice when someone comes and say, I don't really know, I say, well, best economically way to do it and sometimes give you a reasonable power, it's probably to do seed sample per lane on the high-seq, which is about 35 million, okay? Remember, this is RNA-seq, this is not DNA microarray. So you can do 35 million, do an analysis, and then maybe you start seeing something that are expressed at low level, but under pressure you don't have quite the right number to do the statistic. You can go back and run another line of sequencing and get those two data together. That's perfectly fine, okay? You can't do that on a microarray, right? Once you run a microarray on a sample, you can run the same microarray twice, but it's not going to give you twice more data, okay? So that's the beauty of RNA-seq. You can always sequence deeper to get the genes that are expressed at a lower level, okay? So that's why I say, unless you have some very good reason early on to target genes that are expressed at a very low level, you probably are better off to take a middle-of-the-road approach, do your analysis, see what you find, and then make an educated choice, right? And you also depend on things like the biological variation in your groups and all kind of things like that. So having at least an analysis on some level will help you decide, okay?
So you wouldn't have a problem if I was to pool 4 libraries,
Mm-hmm.
2 control and 2 experimental.
Mhm.
And I pool them one day
Yeah.
on one lane,
Yeah.
one flow cell. And I need deeper sequence.
Mm-hmm.
Do I need to prepare then new libraries for those, or do I need to rerun?
I will run the same library.
Rerun the same libraries?
Yeah.
And it's okay that it got run on a different machine, a different day? all
I'm
this
going to
stuff?
get that to that at the end, about
Okay.
the lanes on the machine.
Okay.
I guess to other things like confounding effects and all those kind of things. I won't go into to the end, okay, and what are potential bias. But yeah, I will use the same DNA. I mean, once you have the library, what is the library? It's double-stranded DNA, right? You can freeze it. You can keep it in your freezer for years. You know, as long as you don't thaw it and freeze it all the time, which we all know is pretty bad, but as long as you don't do that, you're safe, okay? It's DNA, it's fine. Okay. That's the other big one, right? How many biological? So let me put that right out. It's useless in RNA-seq to sequence twice the same library on 2 different lanes just because you want to look at potential variation between the lanes or between the flow cell. The variation between flow cell and lanes have been shown again and again to be minimum compared to the biological variation. So spend your money getting more biological sample. Spend your money getting more biological replicates. That's where you should do, that's where your money should go first of all, okay? Number 2, never do 1. Hopefully it's obvious. Personally, never do 2, because how do you compute within group gene variation with 2 samples? You can't do stat with 2 samples. You need at least 3. On top of that, how do you know that one of them is not an outlier when you have 2? When you have 3 and one of them is barely, is obviously very different from the other 2, then you start to scratch your head and say, well, okay, what did I do a day, right? And you have 2, you can't know that. It may be natural variation within your experiment, or it may be that you have an outlier. So 3, by all means, 3 minimum, okay? Okay. Most of the time, cell lines, 3 is enough. Depend on your experimental design, depend on your tissue after that. I had some people at the Jackson Lab who are doing a lot of work on brain, behavior study in mice. And I can guarantee you there is not one mouse, even if they're all inbred, that react exactly the same way as the other one. So there was a lot of variation within groups. So they had to go to 5 or 6 minimum to start to see something statistically significant, okay? Most of the time, I would say 3 is enough, 3 or 4, okay? But again, it depends on your organism. I can't tell you that. And most of the time, obviously, you don't know your within group natural variation because you need data to know that, okay? So you have to make some educated choices there on what is your biological variation and what is the across group versus the biological variability. That's actually an interesting paper from the Nature of Biotech. It's in the supplemental material of that paper, but and it's a good paper to read too. But if you look, they have that graph. And basically what they do is they use a global general linear model, for one of you who knows this kind of thing, and mixed linear model, to tease out what is noise, what is technical noise, and what is really the biological variation. Okay. And when they do that, and they plot, they calculate the biological variance against biological variance plus technical variance. So obviously, if you have 0 technical variance, you get to 100%, right? Okay. If the technical variance is dominating all your signal, then you get to 0. Okay. So they look at sequencing versus arrays, and you can see that obviously in sequencing you get a much better, you get a lot less technical variance than in the arrays. Okay. It was an interesting graph, I thought. And you get basically better, you detect better differential expression. Part of that has that to do with the arrays. Basically, you have a lot of cross-hybridization between props of different genes, and they don't do very well at low-level expression genes as well, and they saturate on high-expression genes. Okay, you never saturate on RNA-seq. Okay, you can always sequence more to get the lower one. So that was an interest. But the bottom line is, you don't know this, but this is what you want. Okay, measurement error is pretty small compared to the biological variability. Okay, the last one is always bigger. Okay, so maximize the number of samples. Don't worry too much about the measurement, with the caveat that I'm going to show you at the end. There are some things that you still have to think about. Pooling, a lot of questions about pooling. I wish you should not fret it too much. It's not that difficult. So pooling is basically multiple sample, right? Every time you make a library, there is some kind of barcode in the adapters, a unique sequence that the machine can read, so that you know that the sequence that your sequence can be linked to that particular barcode that is in the adapter that is attached to it, and now you can relate that back to your sample. So you can put multiple samples in one lane, so it's a lot cheaper, right? Illumina platform, the biggest thing to remember, actually on NetSeq we don't know that, by the way. The NetSeq is a new instrument, it works a completely different way, so it's very open right now. But that is on the MiSeq and the HiSeq. So you have two lasers. One is looking at illuminating the G and the T dyes. One is illuminating the A and the C dyes. Okay, and when he reads the barcode, he really likes to see on a given flow cell, on a given lane, he wants to see a G or a T somewhere and an A or a C. Okay, so if you have two barcodes, you should be attention. You should have a G or T and an A or C between the two. Okay, and so long across your whole barcode length. That's really optimal when you do that. Honestly, we've done barcodes that were not that good, and it worked really well too. So it's a good thing to try to follow this, but don't throw your library away if you happen to have an A, and CC, and then maybe different bases after in your two barcodes. Okay, it will sequence. You maybe have a few more percentage of sequences that will not be able to be assigned to one of your two samples, but you will get something. But if you can, do this. In Illumina guide at the end, in the appendix, if you download it, it's free. You'll have some kind of a number of recommendations. For example, avoid those guys together. They don't work very well. Some kind of other recommendation if you want to pull 2, 3, or 4 together. Okay. Once you go out beyond 4, it's usually not a problem. Okay. There are also recommendations when you use a dual index that you guys did not use. Well, actually, yes, you guys used. On the kit we used, when we lighted the adapters to your DNA, on one side you have a barcode, on the other side you have a different barcode. It's called a dual index primer. Okay? So we actually read barcodes on both sides when we do the sequencing. Okay? The reason is that you can go up to much higher level of barcodes. Okay? When you do that, the plate we use had 96 different barcodes, for example. And I told you this morning we are using another kit with 192. So there are other different recommendations, basically, on how you should mix those, okay, to make sure some of them. Look at it, okay? I'm not going to paraphrase the protocol. Look at it. It's very well done. It tells you, for example, if you want to pull seats, you should probably play it on a, take those seats on the plate, take those seats, and so on. Okay, that's the best design. Okay. If you want to pull, seats here. Dual index, those seats here. If you want single index, which is only one barcode, the same barcode on both sides of the molecule, well, you can take those seats and so on. Okay, so it really gives you really a layout of the plate and tell you should get those together if you want to pull them by 6 or by 8 or by 7 or by 9 or by 12. Okay, so all at the appendix and the end of the protocol. There are about 10 pages full of that. Okay.
One flow cell says index run that you guys ran for us, and the other one says dual index run.
Mm-hmm.
So that first run only has one index?
No, that was a mistake.
So they're both dual index.
They're both dual index, yeah.
Everybody
They're both dual index. Yeah, that was a mistake.
Okay,
But they ended up at the 12 were different anyway, the first 12, so we were able to.
okay.
Yeah, the samples will run as one index, but they actually have the two index.
Right.
So when the run was set up, somebody did
Yeah.
a mistake and put
So,
it
yeah,
one.
on the dual index, as you can see here, in every row, in every column, the first index is different.
Yeah.
And on every row, the second index is different.
Yeah.
So we took the 12 on top, they all had a different index.
Okay.
Okay? But they are all dual index, actually, yes. The 96 plate for Illumina are all dual index, by the way, all of them.
So then you just deconvoluted?
Yeah.
afterwards?
The software is actually, then I was here, plug it into the software, and it basically deconvoluted. Yes, yes. So basically what happened is you read 100 bases, so in your case, we did 150 for one run and 75 for the other, if I remember.
Yes.
And then the machine removes the newly synthesized strand of DNA, bring another different sequencing primer, and now read the barcode. So it's two different sequencing read, completely different. But since we know where it is on the flow cell, we have a physical address for those two reads on the flow cell. We have an SY coordinate. Then you can relate those two and say that 100 base per or 150 base per read was on a molecule that has that barcode. And now I know that that barcode came from that sample, so now I can split those. So yeah. Different type of buys. So those guys, actually, so they send DNA to 7 labs in Europe. RNA, they send RNA, sorry. They send RNA. They extracted RNA and send it to a variety of a bunch of labs. Between 40 and 130 RNA samples for each lab. 5 of those RNA were present, they were sent to all 7, okay? One of those labs did actually run all the RNA that were sent to all the other labs. So there is a lot of cross-references. They all use the same kit, they all use the same machine, they all use PAN 75 sequencing. So they try to standardize it, okay? And say, okay, what difference do we see now between the different labs? Okay. Again, it's a big paper, so I'm going to show you only a couple of slides, but when they look at the 5 that were shared across all the different labs and do, it's not PCA, but it's something similar to it, and they basically sort those out. Each one of those colors is a different RNA sample, and those are the different lab. You can see that you can sort, if you look at exon level now, just exon level, okay, just each exon, how much is it expressed, okay, and you should expect that the different measurement between the different labs from the same gene should get together, right? Because they've done a good job. That's what you see, you know. All of those red samples clustered together, all the different labs, found more or less the same expression. Let's have that one, actually, a little bit. You see, it's an outlier for that lab. That lab did not such a good job, as good as the others. But still, I mean, you can easily differentiate the expression of that gene from the other 5, from the other 4, okay? So I was encouraging, when you go to the transcript level, things get a little more muddy, okay? Part of it is because some of the labs sequence more than others. Some of those 5 common RNA samples were sequenced at different depth between the different lab. So at the exon level, it's usually not too much of a problem. Obviously, they did not pick genes that were expressed at very low level. They put genes that were expressed at kind of a medium range. So you got enough read at the exon level, but when you're trying to aggregate along the length of the transcript now, if you don't have, you could easily have one exon that just don't have enough read in one lab to have to give you a good expression. And now it gets a lot more muddy.
I thought the FPKF or whatever that.
So the FPKF is done to standardize. So it does 2 things, right? Yes, it does that. But you have to remember, let's say I sequence 30 million reads, right? And I sequence 120 million reads. I will get 4 times more reads, 4 times, so you'll get the same FPKM, but the underlying count is 4 times as many count, right? So now if you aggregate everything, those software use that to create those transcripts, use the raw number of count as well as the FPKM, okay? And you can easily miss one, you know, if you have very few of those, you could miss an exon entirely, okay? Some of them lab, I'm not going to show you, but when they made the library ended up having different GC content, even for the same RNA.
Same RNA.
Yeah, it was really interesting. Same kit, okay? So clearly, and most of it is probably the CDNA synthesis, okay? So some of the labs didn't do such a good job as the CDNA synthesis, you know? And so exons that have a high GC probably underrepresented in those labs, okay? So you get a little more muddy here.
You say same kit, but not necessarily same lot number.
No, not necessarily the same lot number, no. And one of the things that I found quite interesting is that diagram here. So they actually try to break out, they use a general linear model again to try to break out where does the technical noise come from, okay? So first of all, the residual contained about, you know, that's biological variation. That's a variation that, you know, that you expect from your biology because they did not part that out. But if you look at it, for example, you look at the real number value. Well, all of them had to have a real number of 8 or above, okay? And they send the RNA to everyone anyway, so it's not like everybody has studied their own RNA. So in that particular experiment, it has very low effect on the total variance because we use the same RNA. But RNA extraction batch explains a lot more of the variance. Even if they send the RNA to everyone, one lab extracted the RNA. They extracted the RNA in different batches, different days, different technician maybe, okay? Quite a bit of the variance come actually from when those batches were, not when, but yeah, different batches of RNA. Okay.
I thought you said they send one pool of RNA to everybody.
Yeah, but one lab extracted the RNA, but he extracted them in different time. He didn't extract the RNA. So they send a lot of RNA, okay, to those, okay? So they had to do different batches of RNA extractions. Hmm?
All from the same tissue, or?
Yes. I don't remember what tissue I'm trying to remember. I don't know. I'll have to look at the paper.
This experiment, it would seem to make sense to pool all those batches.
Exactly.
It would, and they did not. That's interesting. I agree.
Yeah.
That's one of the things. Another thing too is when they look at it, when you see primer index, say, oh, wait a minute, different barcode with different variants. But what happened is that different lab sequence at different length, at different depth. So, and they could mix and match the RNA sample with the index. There was no guidelines there, okay? They could take whatever barcode they wanted for whatever RNA sample they wanted. So what is happening here is that most likely, because there's difference in read depth between the different labs, you see that reverted between the different index, and you see a difference between the lab now, okay? But I don't think it has anything to do with, I mean, in our experience, we have done a lot of heavy pooling, and the sequence of the barcode doesn't matter, okay? It's not, I mean, there are no barcode that sequence a lot better than others, okay? So that is probably spurious. But another one is interesting, library preparation date. So library where each lab makes, each of the 7 makes multiple libraries, obviously, okay? Within each lab, there is a variation when they made the libraries, okay?
Which day is good?
Well, you have to have a full moon, you have to turn 3 times around your building in a clockwise manner.
February 29th.
Friday 13 is pretty
There
good.
you go.
Yeah.
It's today.
They're not good, but we have to be aware of that. Okay. So basically, the bottom line is, you know what? If you have a few samples, extract your RNA all the same day by the same person at the same time. Do your library the same day by the same person at the same time. Okay, try to, okay? I'll talk about that in the next slide, so I don't want to go ahead of myself. But in this paper now, what they did is they spike human and Drosophila, I think, RNA with basically spiking, okay? RNA that do not map at all to the Drosophila or the human genome, okay? And use those spiking to do a number of things, in particular to measure, the magnitude of expression level they could detect, about 6 order of magnitude. They could, use, how good are the replicate between different RNA-seq sample. Now I'm not looking at the RNA-seq itself of the human, I'm looking at the spiking, right? So as a technical variation. Right? So it's pretty good. And obviously you can see that if you have more reads, you have a better correlation, that you start to have much lower number of reads between the 2 replicates. Okay? But Most interesting was those, actually. This is across the length of an RNA, and they measure how much depth. What is the coverage? What is average coverage? And those spiking now, so they know what they're measuring because they added to it. Okay, they know what they have. What is the coverage from your 3' end to your 5' end? You expect, a flat line, right? If everything was perfect. Not at all. Okay, it's clearly very different. The fat line is the average, and the dotted line is a variation around that average. Okay. Definitely, there are some similar, significant biases across, and you can see it every time you look. If you take the chance of just looking at FPKM, you actually look at the genome viewer, you map your read on it, and you look at it. You look at different exons of a gene. You'll see it's all over the place. There are some exons that have 2 or 3 times the same number of reads as the next one. If within the same exon, you'll see large differences in the number of reads as well. Okay, some of that is variation, obviously, but when you start to see 3 or 4-fold difference within the same exon, it's not stochastic variation. Okay, it's something else that make it more difficult to sequence part of that exon than the other. Okay, that's exactly what that line shows. The other thing, so you need to be aware of that. Okay, there are exons that you are not going to be able to sequence very well. Okay. They look at, and that's actually mono. So now they look at 2 different protocols: unstranded and stranded. You remember what I told you about? So unstranded, basically, it was interesting. It was an unstranded, but instead of doing they use a mix of poly-A and random primer to do the cDNA. Okay. The first cDNA, and then the second cDNA, they use a mix of random primer. So random primer are used in both first strand and second strand, okay? And when they look at the sequence, so 0 is actually the beginning of the sequence, okay? But they map that against the genome, since they have the position now, okay? So they say, okay, I'm starting to read my sequence as 0, but I want to know what is the sequence in the genome before and after. And is there any bias? I mean, if you had no biases in the cDNA synthesis, in the PCR, and everything was sequencing exactly the same at the same level, you should see a flat line. Okay, you should basically say every sequence has exactly the same probability to be sequenced, right? You see some variation because you already are only sampling from a population, but since your sample is large enough, you should basically say a flat line. Obviously, you don't. There is for those random primers, you have a very typical, C, for example, here, much larger number of T's here, underrepresentation of C and T's here, up to about 11, base 11. And you know what? Because the random primer are not so random after all. Okay. It's actually something I was described first about 3 or 4 years ago, and there is even a mathematical model out there to account for it. But because the random primer don't really prime that randomly in the genome. Okay, they have preferences. Even if they are truly random primers, some random primers bind with more strongly than others. Okay, and they are more likely to generate a first-strand cDNA than others. Okay, so you will have a differential number of reads and a different ability to detect RNA expression depending if your RNA has those sequences or not. Okay. Second of all, and that reflects here. Obviously, that's one of the major causes of this actually. The other thing, when you do stranded, so now when you do stranded, it's a little bit different between the 5' and the 3', the first strand and the second strand. But you still see clear biases, okay? First strand and in the second strand, okay? So, and most of it has most likely come to the cDNA synthesis, okay? And most of that, actually, when they build a model to try to explain this, most of this explains this, okay? So the sequence of your gene has an effect on how well you can detect it or not, okay? In that particular paper, they have a mathematical model that you can use to basically try to smooth the expression, to try to remove some of that. At least I know of at least of another paper out there, but it's older, that do the same thing. There might be more that I'm not aware of, okay? But you need to be aware of the bias, and you need to make a choice. Either you want to compensate for it or you don't want to compensate for it. It's up to you. But you will have it, guaranteed, okay? Finally this. Any one of those? Yeah, you guys have done microarray before? Yeah? Everything you learn about microarray is also true in RNA-seq. Everything you have learned in your original classes in undergraduate on how to design a good experiment, guess what? It's still true. Okay? Basically, and the first paper is much more general. The second paper is it's more mathematical, okay? But it's calculus, okay? It's not like, it's not like differential equation or anything like that. So I'm sure all of us can follow the second one. It's just a little bit more mathematical, okay? But bottom line is that you have to pay attention to the design of your experiment, right? We are talking about different day of RNA extraction, different day of making your library. Well, if day one you extract all your control and day two you extract all your sample, now you have a difference between those two. You don't know if the difference comes between, because you extracted at 2 different days or 2 different technicians or 2 different lots of the same RNA, kit, whatever, okay? Or if it is true biological variation, you don't know. Now, if you had extracted half and half the first day and half and half the second day, then you can use general linear models, which are pretty easy to make. I mean, any statistical model out there, and R is full of those actually, by the way, which is free, can help you dissect that and basically remove the technical variation to only look at the biological, okay? But you have to pay attention to not, as I say, do all your sample one day, all your control another day. Same thing for the library, right? So you have to think about those things so that you don't fall into that trap. And it's just basic experimental design. I'm not teaching you, I'm guessing most of you, anything. But you need to remember it because it's still true. It's not because we are sequencing and spending a lot of money and getting a lot of sequences that avoid the fact that you need to think of your experiment in a careful way before you do it, okay? Because if you have confounding effect early on, no matter how much statistics you do in the back end, you're not going to remove them, right? So I definitely encourage you to look at those two. The first paper not only deal with RNA-seq, but also with microarray, but it's exactly the same principle. And actually, they had an interesting, this is interesting. This is data from the 1000 Genome Project, okay? It's public, right? And they zoom in one location of the human genome, and they look at the different genomes that were sequenced from 16 to 264 and say, how many FPKM I get? I mean, how many reads, sorry, not FPKM, I'm getting. How many coverage I get in that region? All of them were sequenced at 30 or 35H, so more or less average across the genome, the same, more or less the same coverage, okay? But how much in that particular region coverage do I have compared to the average across the genome, right? And they color everything that were 3 deviation lower or 3 standard deviation higher than the average read count across the genome. Okay, basically, every time you had a read count that was wildly different from the average, they color it blue or red. Look at that. You can see distant pattern. Between those, by the time they were to 230, they basically sequenced a lot more of that region at that time than they did here or here. And this is only genome sequencing, which is 10 times easier than RNA-seq, okay? And they still had a batch effect. Okay? And they all come to the library preparation, down to what we were talking about before, okay? So, think about acting. That's it.
Great. All right, thank you.
I can send you the presentation of Mona, I can send it to you, and you will have the references that I put in, so you can look at it, okay?
Yeah. More questions? Yes, Karen.
Mm-hmm.
Question, I actually
Karina.
have two. One from research standpoint, another one from, doing it for students. So the research one, how low of RPKM you consider the gene not to be expressed?
Never. There are some papers out there that show gene expressed in 1 out of 210 cells, actually, and with biological activity.
Wow.
Now they are doing a lot more. There is actually a single-cell paper, RNA-seq paper this week in Science. But they are getting more and more. Some of those single-cell RNA-seq are starting to come up, paper are starting to come up. The technology is about a year and a half old, so the papers are starting to come out now. And what they're seeing is they are seeing, especially in some, depending on biological system, what you're looking at, but they do see things like suddenly you see expression of one or two of, in 1 out of 2 cells, 2 out of 3 cells, and then somehow suddenly everybody expresses the same thing. Okay? So, even 1 out of 10 can have an activity. So there is, it's up to what you want to detect.
Right, but like if you do this analysis, so do you ever, like I usually do a criteria, like set up a criteria of like, if my RPKM is less than 0.5, sort of, like if my difference is between 0.1 and 0.4, I still consider the gene not expressed and I don't call it as differentially expressed. Would you suggest not to ever?
No, I say you're making a judgment call based on what you know of your organism and your experiment, and you're perfectly correct to do it. But I cannot do it because it's not my experiment. It's again, go back to the prior knowledge you guys have about what you are doing. You have to bring that into the table. I can sequence human genome after human genome sequence. I don't need to know anything about that particular sample. It's a human genome. I will sequence it the same way. I mean, it's very set on how you would do it, okay? RNA-seq is not that way. Okay? You have to bring your prior knowledge.
And there'll be a lot more discussion about this with Molly on Monday.
And so in terms of doing the students, I envision then that, let's say, one group of my students can prepare one RNA sample or two, I don't know, and another group of my students could do another one or two, because I don't think they can all do all 18 or whatever the number that I actually want to be analyzing. And basically what I get from your description is that that's very...
It's not bad, but just make sure that one student don't prepare all the control and one student don't prepare all the sample. If they do 1 and 1 and 1 and 1, then you can use linear models to tease that out. That's why those models are made for, really.
Maybe a good idea is to have them each prepare their own, run them on a bioanalyzer, take all the good ones, control experiment, and pool the good ones. You know what I mean?
And even better, if I then do 3, 1 in common,
Sure.
Yeah.
1 in common between the two.
But don't pull them until you check them.
Yeah.
Okay.
Well, yeah. Well, oh, yes. Don't put libraries before you run them on a bioanalyzer and check them.
And
Yes.
I know what I did before, which I totally get that was totally wrong. I would compare 2 or 3 samples, like control and cold or control and heat or whatever. And then sometime later, sometimes in 6 months, I realized, oh, I really want this particular other test or other stress or something like this. And then I would make the libraries and sequence and compare them all together. And apparently that was not a good idea.
No. I mean, I've done macroarray since 2000, and I got beaten in the back for the same problem, okay? You got to be careful of confounding effect and make sure that you can tease those out at the end, okay? There is a lot of literature on macroarray, by the way, on that subject too, okay?
I guess I'm used to thinking about expression data as revealed or indicated by qPCR, where you normalize to some internal gene
Controls,
control.
yeah.
And all of the RNA-seq analysis, differential expression is considered, at least in my experience here, has been in terms of reads per kilobase or reads
Yeah.
per transcript.
Yeah.
Is there,
Well, first
are
of
those incompatible means of doing the differential expression? Is
first of all,
normalization illegitimate for RNA-seq data?
nobody has really done it for the same reason. I think basic reason is that how can you make sure that that particular gene that you're looking, and actin vary between cells, by the way, I've done it. How make you sure that your control gene is really a good control gene? It comes by the same thing for
You
qPCR,
have that
right?
problem every time you do qPCR.
Right.
And that's, you're going to do
Right, and so it's not impossible to do with RNA-seq. I've not seen many people doing it, to be honest. People prefer to do the FPKM, and now they normalize to the 75 percentile and all other type of statistical treat to basically remove the effect of the very largely expressed RNA and stuff like that. But yeah, people tend to stay away from it. What bigger story is that?
Well, it's just another divisor.
No. So it's not necessarily a bad thing, but if you look at it as RNA-seq as an explanatory tool, and you are going to choose the qPCR after that anyway, it's probably better to shy away from internal control, from what you think is an internal control. There are some spiking. So the last Geon Emerald, one of the last I show, they use spiking, and you can buy those spiking from Ambion, I think. But it's $100 a library, so it's basically double the cost of making the library. So pretty much nobody uses it except on Tom Gingeras here, and very few overlap because it's too expensive. But that's obviously the best way, because now you have a spiking, you know exactly what to look for, right? So you know, that's another potential issue. I mean, a potential solution. Yeah, Ambion is they sell it for. Yeah, I mean, the best way would be spiking. Personally, my personal view would be spiking something. I've done it for my career as well, and I think it works much better.
You finished, Bill and Eric? We reserve the right to email you. Thanks again.
No
Eric. That
problem.
was really great. Thank you.